healthy aging, and how it impacts aging in disease states like Alzheimer's disease. So I studied cannabis. The reason why I study it is because it is increasingly accessible. There's increasing use, and we still don't have super clear guidance on the consequences of cannabis exposure, whether it's, across the lifespan, starting when you're older. We know a lot about adolescent exposure, but we know a lot less about things like perinatal exposure or aged exposure. So what I'm showing you here is the number of individuals in the United States who are reporting that they are daily or near daily users of marijuana, the green line, or alcohol, which is the red line. I'm gonna go ahead. Oh, I can't get it to turn on. Bummer. So what this is showing you is that for a prolonged period of time, alcohol was by far the most commonly used substances between the two of these, and that its usage rate, was climbing sort of slowly. This is in contrast to cannabis, where you can very clearly see that as decriminalization, legalization, increased availability of both recreational and medicinal marijuana is rising during the early 2000s into the aughts, we are seeing a commensurate rise in the number of daily or near daily users. And importantly, in 2022, for the first time, there were more American adults saying that they were daily or near daily marijuana users than alcohol users. So this is a huge population that we are being impacted by cannabis exposure. And this is across the lifespan. So what I'm showing you here is data from 2013 and 2022, actually broken down by age range. So the black line that's kind of in the middle is showing you any American adults 50 or older, what percentage are reporting cannabis use. And so what you can see is a steady increase in the utilization of cannabis in this population. And then this graph further breaks down by age group, 50 to 54, 55, 59, right? The percentage of that population who are using. And importantly, there's two key things to see on this. The first is that the rate is rising overall. And the second is that we have a good number of users in this middle to early aged group, right? This 50 to 64. But the 65 and plus are just going to continue to catch up over time. So cannabis use is increasingly prevalent across the lifespan. It is increasingly prevalent in older adults, and we still lack clear guidance on the consequences of cannabis exposure in aged or diseased populations. So cannabis is referring to the whole plant, which contains multiple what we call cannabinoids, polyphenols, terpenes. Some of them are psychoactive, some of them are not. I am going to focus specifically on delta-9 tetrahydrocannabinol, or THC. So THC is the primary psychoactive constituent of cannabis. It's what makes you high, okay? And this is in contrast to something like cannabidiol, which is considered non-psychoactive because it doesn't induce the high feeling, but it does alter behavior in interoceptive state. It's more responsible for things like the anti-nociceptive effects, the pain relief effects, the relaxation effects, the appetite effects, right? All the sort of peripheral system is cannabidiol. The vast majority of the brain stuff is going to be THC. And as a neuroscientist, I'm a brain stuff person. So THC acts as a partial agonist at the cannabinoid receptor 1, CB1 receptor. This is part of an endogenous cannabinoid or endocannabinoid system. And THC is a partial agonist at CB1, and this is in contrast to CBD, with cannabidiol, which is activating a different receptor, the cannabinoid receptor 2. And CB1s are one of the most highly expressed G protein coupled receptors in the nervous system. So G protein coupled receptors are receptors that when they're activated by their ligand, they activate different intracellular signaling cascades, so they can have really diverse effects on cellular function. And activation of this CB1 receptor, it's highly expressed in multiple brain regions because it's critically important for sort of short-term tuning of synaptic activity. The endocannabinoid system includes the CB1 receptor that's on the presynaptic side of the synapse, so the one that's releasing neurotransmitter. And the endocannabinoid, the endogenous agonist, is produced in the postsynaptic side. and it's produced under conditions of very high activity. And so then what it does is go back and reduce the amount of neurotransmitter release from the presynaptic terminal. So it's really important for sort of short-term gating of how much neuronal activity there is. It is also strongly implicated in longer-term forms of plasticity, and particularly in a brain region called the hippocampus, which for our AD and dementia folks, definitely, I imagine we have a shared interest in that region. Important for lots of forms of memory, contributes to mood, is one of the regions that is degraded during Alzheimer's disease. And so our focus is going to be on THC activity on CB1 receptors in the hippocampus, because this is where they are richly expressed. They are contributing to behaviors relevant to aging, addiction, disease, and they are critically important for tuning both acute and long-term functions. The endogenous ligand here is anandamide. I'm not going to talk about it very much, but it's just to set up this idea that this system has an active role ongoing in controlling neuronal activity. And so what we are doing when we're bringing in exogenous substances is we are changing that level of activity. And interestingly, which may be related to some of the effects I'm going to report in a minute, the endogenous ligand for the CB1 receptor, anandamide or AEA, there's evidence that it's actually reduced during aging. So whether that is a reduced capacity to produce the agonist, whether that is an overall reduction in the level of activity, meaning less signaling is required from that system, is not super clear. But I think it's interesting to consider this when we see some of the data that I'm going to present later, this idea that the aging brain may be depleted or have a lower level of the signaling molecule, therefore leaving space for the exogenous to actually change behavior and brain function. The reason why I focus on THC is because it's the high, that's what makes you addicted, it's acting on the receptors that are in the brain, it's involved in plasticity, and importantly, modern products that are coming out now have incredibly high levels of THC. And increasingly, they have only THC. So particularly in the recreational market, the point is the high. And so you're going to see a huge variety of products with ever increasing quantities of THC, meaning we have more users and they're being exposed to more THC than they would have historically if they were using plant in the 1970s or 80s. Okay, so plant is being cultivated to have high THC, 10 to 30% in the raw plant format. Vapes, which are an increasingly common form of administration, we're starting to get up to 95% THC, the same thing, high levels in these concentrates, right? So THC is like 10 times stronger now in just plant, let alone all of these special products that further concentrate and enhance the action of THC. So lots of people, lots of exposure, unknown consequences. So the knowledge gap that we're trying to address is, what does this do to the aging brain? Okay, so from human studies, we already know epidemiologically that there's an increased use in older adults for both medical and recreational settings. For a number of disease states, drobenol, which is a cannabinoid similar drug, or even just cannabis itself, is being recommended to handle things like chronic pain, insomnia, decreased appetite, anxiety. And from recent studies looking at scans and tests of middle and older adults in the UK, what was found was that there's a moderate chronic use is associated with larger brain volume and improved cognitive function. So unlike what we might be predicting, which is a negative impact, here at least they're finding a positive impact. Importantly, moderate, meaning low total amount of use, like how much cannabis they're taking in each time they take cannabis, but chronic, meaning they have been taking that level of cannabis for a prolonged period of time. And the reason why I point that out is because those are really two critical factors that help us understand the discrepancy in the literature between whether cannabis is beneficial or deleterious. So brain function wise, there may be a benefit, but if we look at chronic cannabis users and we look at their oral microbiome, there's evidence of dysbiosis, and particularly in what we're going to call pathogenic species, ones that make products that are neuroinflammatory, ones that may translocate into the brain and alter neuronal activity. So these cannabis users, although these were an adult population, not an age population, it indicates that in addition to central effects, we could be having peripheral effects that could then subsequently influence brain function. Human studies are great, but a lot of confounders, right? I'm a preclinical person because I'm kind of a control freak. And with the human studies, I think one of the reasons why we don't have super clear guidance is because it's very hard to control for, geographic region, other usage, nutrition, socioeconomic status, education, all of these other factors that we know are impacting some of the different features that we're measuring. So in animal studies, we've found some evidence that aligns with a potential beneficial effect. So chronic low-dose THC reversed age-related cognitive decline in a normal aging population of mice, and in an Alzheimer's disease, the 5XFAD mouse model. At the same time, our group found that if you give repeated high-dose T exposure to aging animals, you disrupt spatial pattern separation. So spatial pattern separation is a particular form of episodic memory. It's mediated by the hippocampus. It's actually one of the first sort of behavioral changes that you can detect in humans with Alzheimer's disease. And so that's why we were really interested in this idea that, we're producing a deficit in this AD and aging related behavior. And then finally, if we take the microbiome, the changes that were found in the human cannabis users, and we introduce one of those specific species, Actinomyces mayeri, to a mouse, what we find is an immune response, evidence of neuroinflammation, macrophage infiltration, all these signs that the brain is not feeling good, right? So again, sometimes it's beneficial, sometimes it is deleterious. The effect could be caused directly by THC. The effect could be caused by changes in peripheral systems that ultimately feed back onto the brain. So animal studies are great because I get a lot more control, but the downside is that they oftentimes use routes of administrations or formulations of the drug that are really inconsistent with the way humans use it. And pharmacokinetics, pharmacodynamics is really important for the development of addiction-like behavior, the amount of drug that a person is going to use, the type of plasticity that is induced, right? So having more translationally relevant routes of administration in our animal studies are going to help us to, again, further sort of control and have a more translational model. And so to address this, our group is using a vapor model. So what I'm showing you here is a little vapor chamber. That little vape pen is meant to essentially indicate that vapor enters the chamber. This is a sealed chamber. We essentially fill it with a set amount of vapor. The animals sit in it for a certain amount of time and the vapor is cleared. And we're gonna do cycles of that, multiple trials a session, multiple sessions over time. And again, we think this is a great model because vaping is ever increasing, right? I don't know about you guys, but I see those vape clouds all the time in people's car, at the bus stop, walking around, right? And well, most of that is likely nicotine. That is also a very popular delivery strategy for cannabis. Right? So again, like I said, route of administration really matters. Our goal here is try to get something that's has a bit more face validity for what humans are doing, knowing that inhalation versus injection versus IV are all going to change the animal's experience of this drug and the consequences of exposure. So more translationally relevant delivery system. So what we're using is a high THC cannabis extract. So this is from plant, it's not synthetic. And essentially what it is a plant that has been raised to have very low CBD and very low other cannabinoids and be primarily THC of all the chemical constituents, right? So remember, the remaining 30%, less than 1% of it is these other cannabinoids. The rest is terpenes and polyphenols and other compounds, right? So ours is a nearly 70%. And what we do is then dilute it to 100 mg per mil of THC in our vehicle. And importantly, our vehicle is propylene glycol vegetable glycerin. This is the same vehicle that's used in human vapes. We're using it at essentially the same ratio. And so again, getting at that translational relevance. So you've got the vapor chamber. We can computer control how much puff and how long for the vacuum, right? Also depicted here, but not used in this study, is that these chambers are set up for volitional taking. So the animals can learn to like poke in a hole and get a puff. And that's important for some of our other studies, but is not employed in what I'm going to talk to you about today. And so key variables in this model are, you know, the number of puffs, like how many total puffs are you getting? How long the puff is delivered, right? If it's a 1 second puff, you're getting a third as much vapor as if it's a 3 second puff. So these are the ways that we alter the sort of dose, but it's not dose. We're going to call it exposure. And then how long are they sitting in the vapor? And how many times are they going to be exposed to the vapor? So for each study, I'm going to define those different variables. All right, so behavior is a key part, cognition is a focus. And really for this study, we focused on 3 forms of cognition: working memory, recognition memory, and spatial reference memory. So these vary in the, you know, sort of duration or persistence. Working memory is really when the animal's in the task, they need to retain that information to complete it. We're going to test that with something called the Y maze that I'll explain in a second. Recognition memory is a more long-term form of memory. It's episodic. We're remembering an object that we've seen before, whether we're familiar with it or not. And we tend to use short retention novel object tasks, meaning the animals learn about the objects and then they're tested shortly afterwards. So, well, this can be a really long-term memory. We're typically doing it within a 24-hour or less window. And then finally, spatial reference memory, which is a long-term spatial memory. We're testing this with Barnes and Morris ways. And I'll talk about those in a moment. What I want to highlight that we didn't do in this pilot because of the scope is that there's a number of other behavioral assessments that we can do in rodents that are very relevant to the aging and the Alzheimer's population. And these are in the categories of social, affect, and motor tasks. Okay, so animals do social interaction. They would prefer to interact with another mouse than another object. And we already know from prior studies that withdrawal from drugs, stress, disease states can all reduce this natural preference, limiting the amount of social interactions that the animals are getting. And from a translational perspective, we all kind of, it's pretty well established that social connectedness is a key variable in healthy aging. So if some drug exposure is reducing social interaction, it may be amplifying the negative effects of a disease state. They can also do anxiety-like behavior and anhedonia-like behavior. This is just, you know, how willing am I to explore something? How much do I want something good? Again, both very important for our human populations. And then finally, motor function and learning. So how much are they moving around? How coordinated are they? How well are they planning for complex movements? We can test for all of these things as well. So you can see that in this pilot, we have a very limited focus. But the goal is to expand this into a number of behaviors that are relevant for the population of interest. So in our first study, we asked how female rats, how the oral microbiome and their cognitive behavior is changed by chronic THC vapor exposure. So these are retired breeder rats, so they're all females, and they're greater than 220 days. So these are more like middle-aged. So that is one of our limitations. And you can see, you know, the specific details, but they're basically getting chronic, what we would consider moderate to high dose exposure. And then we're going to test the animals for behavior before and after the vapor exposure to see whether it has induced a changing. And we're going to collect oral microbiome before and after to see whether the vapor exposure is sufficient to change the oral microbiome. Okay, so the first task we use was spatial location recognition task. This is the pattern separation task. I want to make sure I'm looking at time because I want to make sure I get to everything. So just briefly, in this task, animals learn about 3 similar objects, the same object. What they're learning is their position in space. So what's depicted here is the rat in the middle, the 3 identical objects in the arena, and just outside the arena, the animals have proximal cues that they can use to remember the position of these objects, right? And so the task, the memory, is tested in a this shifted paradigm. So what we're seeing here is animals are learning about the objects, their positions, and then we're coming back, in this case, 24 hours later. And what we've done is one object has remained in place, stationary. One object has been removed entirely, and one object has changed position. And what happens normally in this task is animals are going to spend more time interacting with the moved object because something has changed about it. The other object is exactly where they expected it to be. This one is now somewhere else. So they spend a lot more time exploring that object. So what I'm showing you here is performance based on the percent of time interacting with either the stationary or the moved object. Vehicle group is kind of this purple and blue. THC is green and orange. And importantly, there's a few things that we see here. So first, I just want to focus in on the vehicle. Prior to vehicle vapor exposure, the animals that were assigned to this group, importantly, assigned to this group before we ran the behavior, which is a lesson learned, showed normal SLR performance. They spent more time interacting with the moved object. And interestingly, when we tested them post, this preference has shifted. So this, if it was a properly powered study, there's no stats here, this is pilot still, would be indicative of a deficit in spatial location recognition memory as a consequence of simply just the vapor exposure itself. If we look to the THC, we have a little bit of a problem, which is that the animals that happened to be randomly assigned to the THC group were apparently worse at this task than their neighbors. So they're starting out with a deficit, which means we can't interpret a post effect. And so from now on, what happens is animals get screened and then they get assigned so that we're ensuring that the groups are balanced. This was very unexpected because we're typically, this is a pretty consistent performance. So we were really surprised at this huge group difference. We thought, okay, well, maybe the task is a little bit too difficult. So the next group that we ran, we used a modification, this novel object recognition task. This is episodic memory. Animals are learning about the identity of two similar objects. They're coming back 24 hours later. One of the objects is new. They're gonna spend more time with the new object. And what we can see, again, vehicle pre is in purple and post is in blue. And we're seeing that group, which seems to just be the smarter group, doing normal novel object performance and showing a deficit in novel object performance in the post. And again, because our groups are unbalanced, we didn't really get a great effect here in our novel object performance. But the one thing I do want to say is it certainly looks like vapor exposure itself may be impacting performance, but we're going to flesh that out in subsequent studies. So then the last question is, does it change oral microbiome? We're going to sample. This is really important because the oral microbiome, much like the gut microbiome, has a great ability to alter physiology, including brain function, right? So if we decrease the amount of commensal or good bacteria, or we increase the amount of pathogenic bacteria, we are essentially in a state of dysbiosis. And oral microbiome dysbiosis has been linked to, obviously, oral health diseases, but also vascular diseases, exacerbating existing immune dysfunction like in diabetes, cancer, and Alzheimer's disease, right? So whether it's them moving into the brain, direct translocation, or it's them changing what they're producing, there's evidence that changes in the mouth can influence the function of the brain. And that prior study showed us that in humans, chronic exposure was favoring an increased abundance of a pathogenic type of microbe. So what we were trying to ask is, do we see the same effect in our animals? So what I'm showing you here is the first measure we looked at, which is alpha diversity. This is how rich is the sample? How many microbes are in there, right? This is a within group kind of measure of richness. And it's telling us about variety and abundance, but it's not taking identity into account. So it's literally just like how many. And of that, how many big groups are there? And so observed is just a total count. Shannon and Simpson are both sort of mathematically adjusted to account for not just the total number that we see in observed, but their relative variety and abundance. And importantly, regardless of which metric we look at, I think the most obvious effect is that whether it's THC pre to post, which is orange to green, or vehicle pre to post, which is blue to purple, the vapor exposure itself is altering alpha diversity. So this isn't the ideal measure to ask about how similar or dissimilar groups are from each other. To do that, we're going to look at beta diversity. So this is essentially saying, of all the bugs in the mouth of group 1, how many of them are also in group 2? How similar are they? And in this PCOA plot, which is actually like a multi-dimension that's been flattened, the closer points are together, the more similar the species are between those groups. So what I'm showing you here is all 4 groups. THC is orange pre, green post, blue pre, purple post. I think one of the things that you might be able to pick up right away is, boy, do those post groups look more similar to one another, right? They're very closely related to one another. And if I try to simplify it, and on the left here, I'm just looking at THC pre versus THC post, they have some similarity, but they're pretty dissimilar, which is what we expected. We thought THC would change it, right? But if we look at THC post versus vehicle post, it sure looks like the vapor might be driving much of the similarity between these groups, right? And so again, increasingly in these kind of pilot studies, the vehicle itself is doing something. So that tells us overall how similar they are, but it doesn't let us get at individual microbes. And we know from the prior study that changes in specific families are going to be more meaningful than just more overall or less overall, right? And so this is a strategy that looks at relative abundance, essentially what is the most different microorganism between groups. And here I'm showing you all the groups, and you can see that there are a number of changes in the relative abundance of multiple different types of microbes. But if we try to just simplify that to really look at our question, if we look at THC pre versus THC post, in green, what we're seeing are species that were more abundant pre-vapor than post-vapor, indicating that they either decreased in abundance or were overtaken by a new microbial population. In the red, what we're seeing are species that were more expressed in the post than the pre, again indicating a shift towards a changed microbiome. So all of this work is being done with the Forsyth microbiome core, and we still have a lot of work to do to look at the different types of bacteria, whether any of them have been linked previously to pathogenesis and factors like that. So more to come with this. But I think the key take home here is that the point of the pilot was to see whether THC vapor exposure could change behavior and change the microbiome. We have at least preliminary evidence that that is the case. And then the last thing that we did was just look for inflammatory markers, right? One of the ideas is bad microbes increase, they produce nasty products, the brain gets upset. So in this case, we took the hippocampus out, we isolated all of the RNA, so the gene expression, and we essentially quantified how much there were of different genes in our 2 groups, vehicle and THC. And for this, I'm just showing you a couple of markers, an astroglial marker, astrocytes when they are activated or recruited to tissue damage or injury areas, tend to increase their GFAP expression. IL-6 and TNF-alpha are both pro-inflammatory cytokines. And importantly, we don't see any evidence of changes in inflammatory markers. And so this is still ongoing. We have a lot of sample left and a lot still to run. We still want to look at microglial activation. And then we want to start looking at neuronal function and activity. So neuronal activity, cannabinoid receptor expression levels, glutamate and GABA balance, right? Excitatory, inhibitory. And importantly, bring in an air control. Because one of the key things that we learned from that first pilot is that vapor alone seems to be able to do quite a bit. So in the interest of time, I'm just gonna move through this quickly, which is to say, THC vapor exposure alone causes changes in microbiome. It may be causing changes in the cognition. We just don't have a clean enough experiment to say that. We're going to do it different. We're going to do a separate one. And we don't see evidence of inflammation, at least in the sort of small number of targets that we've looked at. We know that the behavior was problematic for one of any number of reasons, including the fact that retired breeders basically never get handled. And so even though we do a ton of acclimation, they are still super stressed out. And the within animal variability of that level of stress might be part of the reason why our groups were so dissimilar, despite the fact that they were the same when we assigned them. They also had different levels of sexual experience. So they had repeated pregnancies, but we don't know how many each one had. So that's a potential confound because hormonal changes like that are going to have lasting changes on brain function and behavior. There was also a high variability in age. These animals were all older than some age, but they weren't all the same age. And so when we're trying to optimize assays for aged populations, which oftentimes require us to decrease the size of the arena because they're a bit hypoactive, or decrease the duration of the retention interval to make sure that the control animals can actually remember it, making the tasks a little bit easier. And so that is a problem. And then more than anything, air controls. So at the same time that we were doing this, we were running a similar experiment in mice. So in this case, we're looking at 11 to 13 or middle-aged male mice. They are getting essentially the same level of vapor exposure as the prior experiment. So this is a moderate to high dose THC in a chronic 20 consecutive days. And again, we are going to look at cognitive behavior pre and post vapor. So in mice exposed to chronic sort of moderate dose THC vapor exposure, what we tested first was spatial working memory in the Y maze. This is the Y maze. The idea here is that the mouse is more likely to go to the arm that it explored least recently, right? Because what it's trying to do is assess how its environment is changing. And so it's in one arm, it has a chance to explore, it goes to another arm, it explores that one, right? An animal with normal working memory is then going to go to the third arm that it hasn't visited yet. And it's going to do kind of a series of moving between the arms that we're calling spontaneous alternation. And what we find is no difference between the groups pre or post on this working memory task. So then we use the Barnes maze, which is a spatial reference memory task. Not depicted here are cues in the environment that are helping the animal to locate that little escape box. So they need to learn where the escape box is, because they're under very bright light and they would really rather not be on the maze. Okay. And what we find here is over consecutive sessions, the THC and vehicle animals are performing similarly on this Barnes maze, indicating that at least in this spatial reference memory test, there is not a deficit. So the Barnes maze is stressful, but it's not a super high cognitive demand. And so one of the things that we did was actually come back and test reference memory using something called the Morris water maze. So again, not depicted are cues in the environment that allow the animal to learn the location of that little platform. Mice can swim, but they don't love it. So they're under a lot more pressure to get out of the Morris water maze than the Barnes maze. And so this Morris water maze has a higher level of stress and a higher cognitive load than the other task that we ran. And interestingly here, what we find is a beneficial effect of THC. So on the y-axis is how long it takes them to find the platform. The faster you find it, the better you're doing on this task. And what we can see is that over time, both groups improve their performance, but the THC exposed animals who are not high when they're taking the test, but they had prior exposure, are performing better on this task than the vehicle controls. And this was really exciting to us because it actually produces a result that's consistent with a prior study. So in this study, this is one that was using a mini pump. Again, the latency to find the platform is the y-axis. Time showing them learning the task is on the x. The vehicle group is the solid line and the THC group is the dotted line. And so this study used a different formulation, pure THC at 3 mg per kg. They used a different delivery method, an osmotic mini pump versus vapor. The animals were a similar age, the amount of exposure was similar. And here we can see both studies are showing this beneficial effect of THC. So it seems to be enhancing performance on a reference memory task with a high cognitive load. This to me is sort of like telling us that we're at a threshold of exposure. Whether it's we need less THC or more THC, we're at this point where only when we really push the animals do we see the difference. So that probably means that the effect is sort of weak but present. Whatever the change in the neural activity is. So THC vapor, really, we can see it enhance performance in memory tasks, but only under conditions of high stress and/or cognitive load. So we have some subsequent studies that are going to try and disentangle that. We find that the vapor model in mice is recapitulating a prior study, which is great because totally different routes of administration, different formulations, but a consistent result. But this study, we really only did a limited number of behaviors, and we only used males. And they were aging, but not old. So what we're doing now is from these animals, we're looking at the molecular consequences of exposure, asking what has changed in the neurons in these animals as a consequence of THC. We're doing a functional assessment using electrophysiology. So this is a strategy where we take a slice of the brain, in this case hippocampus, out of an animal. It's called an ex vivo preparation. So the tissue is still alive, the neurons are still connected to one another, and they are still firing. And then what we can do is use very sensitive electrical activity measurements to literally measure the communication between neurons, the how much depolarization occurs when there's a glutamatergic synaptic release, or the magnitude of plasticity if we strengthen or weaken connections between specific neurons. So this is a really great acute ex vivo way to ask, did we change how this circuit is working? And could that change explain why we're getting a behavioral consequence? And then the next piece is really to bring in some of those additional behavioral domains. The ones that I talked about that were relevant to this population, but not tested in our prior ones, but also ones that will really help us nail down this cognitive demand versus kind of affect and motivation explanation for why we see it in Morris, but not in Barnes. And then adding age-matched females, right? I think this is particularly important. We know age of onset, prevalence for Alzheimer's disease is greater in females. And so we have these studies underway. And in the far future, we really want to push the age of the animals. And it's an opportunity for us to consider whether we want to bring in AD models to see if we can replicate kind of the same findings that they found in the 5X8 FAD or other disease models. So the last thing that we did, which is not really related to the SC-ADRC project per se, but I think it's of interest because it includes an age element, was that we, in our rat studies, looked at changes in the brain lipidome following chronic THC. So these are middle-aged animals. They had this chronic group had multiple rounds of exposure across development, so a lot of experience. And this is the paradigm in which we previously saw cognitive deficits if immediately following the exposure. So we know that this is a paradigm that's able to modify cognition at least within 72 hours of the end of the exposure. So lipids are particularly interested because, especially for the nervous system, like they are everything. They are the cell membrane. They are substrates for energy production. They are signaling molecules, right? And I am not a lipid person. My collaborator, Dr. Andra Albayram, is much more knowledgeable about this. But I think there's ample evidence that aging and diseases of aging, such as Alzheimer's, alter lipids, right? So if we change the lipid bilayer constituent, we can promote A beta accumulation by limiting access to the degradation enzyme. If we are losing myelination, which is ultimately just it's fat, right? That is helping to maintain axonal integrity, that would alter circuit activity. A ton of different families of lipids have been previously associated in human studies with the severity of the disease. And this is because these lipids are involved in all of these different cellular functions. So it might be of interest to see if THC is actually changing the lipid dome in a way that is consistent with a pro-aging or an anti-aging effect. Okay, so we have a number of groups here, and there's a lot of data, so I'm going to try and really focus this in. So what we're looking at here on the right is, on the left is a whole bunch of different lipid classes. These were detected using LCMS with the Emory Metabolomics and Lipidomics core. So we sent them our brain samples, hippocampus, from exposed animals, and they helped to measure the relative quantity of each of these lipid species. Across the top, we can see our different groups. So we have two sexes. Females are the left set, males are the right set. And then what they've done here is they're essentially normalizing for how does it change compared to the vehicle condition. So in acute treatment, what changes versus the vehicle in a female? In the chronic, what changes versus vehicle in the female? So the reason I'm pointing this out is because our ability to interpret differences and stuff is really dependent upon how we set up the analysis. So this is designed to tell us, how did chronic treatment alter the male or female brain lipidome? And I think, interestingly, what we see here, obviously there's lots of changes. I'm boxing in the chronic group because this is the group that had lifelong exposure. They're the older animals, right? The first thing that we can see is that males and females are different, right? So drug exposure is changing the lipidome in both groups, but not always consistently. So if we were to look at OPC and OPE, which I'm sorry, I don't have a pointer, but they're right in the middle there. They're aqua kind of colored. What you can see is that there's a significant decrease, the darker blue colors in females compared in THC treated versus vehicle. But there are very minimal changes in the males. Those lines are very light. Each line is a different species. There's not a lot of changes there. The other kind of pattern that we might see is exemplified by the ceramides and hexaceramides. So they're down closer to the bottom. They're gray. And what you can see here is that they're having weak effects in both groups, but opposite effects in males and females. And then the last one I wanted to point out was this phosphatidyl serine. This is PS. It's the yellow just above OPC. And these are changing in a similar direction. So they are up, a lot of the species are upregulated in both males and females. But if we look at the history of the animal, what we can see is that the pattern of how they change is very different, right? And so what this is telling us is that drug changes the lipidome, and it appears to change it in a sex-dependent manner, right? So this is a focus of THC within sex that I'm kind of extrapolating these patterns from, but we can test this directly. So what this is looking at is THC effects between sex. So lipid classes on the left, exposure on the top, but now what we're quantifying is the difference between males and females. So this is capturing our sex difference. And those same species that I was pointing out in that other one, now we can actually see, okay, what is the difference between males and females? So as we kind of thought from looking just generally, we've got upregulation in males compared to females for OPE, a downregulation in males compared to females for OPC, so different patterns of changes. If we look at our ceramide and hexaceramide, again, we can see sort of upregulation in the females. So in this case, it's male versus female. So if it's a bluer color, that means there was more of it in the females. And if it's a redder color, it means there was more of it in the males. And then if we go to the PS, we can see that there was minimal difference between the sexes. But we already know that it was changed by THC. So again, this is a deeply bioinformatics heavy analysis strategy, but really what I'm hoping that you take away from this is that THC exposure can alter the brain lipidome in lasting fashion. It's targeting a wide variety of lipid species, so not just the endocannabinoid system. And there are important sex differences, which may relate to changes, the differences that we see between males and females in addictive liability, persistence, risk of relapse, things that we're interested in with THC. The other piece that's loaded into here is age, right? The acute animals are much younger than the chronic animals. And so another dimension that we still have to look at is age-related changes in the response to THC. So lots more work to do. I'm really trying to just show you our pilot data so you can kind of see where we're going next with this project. So age-dependent changes in response to THC, sex-dependent changes in response, lots of different types of patterns. One of the kind of overall things that we took from this data was that males tend to have a lot of changes early in exposure that plateau as they have chronic exposure, whereas changes in the females continue to accumulate over time. What I mean by that is in the acute group, let's say lipid A is up in males and it's up in females. And then in the repeated group, it's up in females, but not up in males. And in the chronic group, it's not up in males, but it's up in females. And what that's telling us is the females are continuing to change where the males are not. So the female lipidome in our study is much more dynamic than the male lipidome, but ultimately both of them are impacted by THC exposure. So more bioinformatic analysis, getting into the THC by sex, the THC by age, the sex by age, lots to do. You know, we need to replicate both of these studies. Which one is it going to be? Mouse, rat? We're going to go mouse. And we can talk about that in the discussion. We've got to add an error comparison to every single experiment that we've done. So, you know, the fun part about pilots is you figure out all the ways that your design was bad and all the things that you forgot about. And it gives you an opportunity to make a stronger experiment, right? And then again, using bioinformatics to get pathways to try and link what we know already about lipid changes in aging and disease to the lipid changes that we're seeing in our THC treatment. So I did not help clear up our understanding, right? Because what we found is in a prior study, we found that a high dose is inducing cognitive deficits. A moderate dose is having beneficial effects on cognition. All of the different changes that we have looked at, whether it's you know, the behavior, the lipidome, what we see is that depending on how much they got, how long they got it, what sex they are, what age they are, these are all intersecting to influence THC impacts. So again, just we need more controlled studies with very specific hypotheses to ask, you know, how does THC influence cognition in X animal at X time with X dose? Our behavioral analysis in aging animals really requires refinement, and we want to expand the phenotyping. What do I mean by refinement? Again, as animals age, their sensitivity to novelty, their motivation, their locomotor output, even their sensory function is decreasing. And so we need to optimize assays for aging animals so that we can actually detect differences between the groups. THC does influence the microbiome, but it actually might be mostly driven by just the vapor itself, which I think has pretty important implications for vaping in general. And then, while we don't see evidence of increased inflammation, there is still this question of how it's impacting neuronal function. So, you know, activity markers, gene expression, and functional assessments using electrophysiology are really our key next downstream steps. And again, we, in the moderate dose, are finding beneficial effects in cognition, but we have not tested a huge variety of other phenotypes that would be relevant. So I'm not willing to tell you that THC is beneficial for cognition, right? But at least in that task, in that age, in that dose, it is. So yeah, follow-ups, female subjects, taking into account estrus, because we know that reproductive state can influence behavior, lipidome, inflammation, neuronal activity. Those are all controls that we're going to need to include in the large study that's underway. And then increasing the age of subjects, older and older, really trying to get to that 16 to 18 month time point. So we're ordering older animals, we're aging them in the colony, and we're trying to get to more of that 60 to 70-ish age in humans. And again, the biggest thing for this is air controls. Every study that we do now has an air control, and it's because there is a lot of consistency in the vehicle and the THC groups to one another, but there's also enough evidence that the vapor alone is doing something that we really need to characterize what that is. Because again, for THC users, for nicotine users, that is a relevant set of information. And so I want to just acknowledge everybody. So members of my lab include my graduate student, Lainey Kimball, Caroline Limbaker, and Kev Delgado. We collaborated with the Forsyth Oral Microbiome Core, Dr. Wei Zhang and Dr. Douglas Johnson on our microbiome study. In study 2, we collaborated with my collaborator, Andre L. Bayram, and his postdoctoral fellow, Etta Karakaya. And in study 3, Lainey did a lot of the work. Andre is helping me understand what Emory is telling us has changed. I want to acknowledge the Alzheimer's Disease Research Center pilot funds, as well as our NIH and our NIDA U54, our mouse behavioral phenotyping core, which helps us expand our behavioral repertoire for our studies, different research groups and different agencies that are helping to supply us animals and reagents to conduct our studies. And with that, I am open to questions, and I appreciate your your attention.
Thank you, Dr. Penrod, for your presentation. So we'll now move into the question and answer session. If anybody in the audience has a question, please feel free to unmute yourself and ask Dr. Penrod.
Thank you so much for your presentation. I'm just curious to know, and you kind of brought this up, thinking about the ages of your mice and the project, especially from study one, had a variety of ages, and then even thinking about the things you were seeing in terms of the vapor exposure itself being detrimental and whatnot, and the fact of looking like from a translational side that the greater use of vapor use, and what might be like the critical part of the life course, particularly potential cohort differences we may be seeing in terms of seeing the results you're seeing with vapor use, even THC, considering you said the use of vapor is giving you a more heightened dosage of THC. So I'm just, from a theoretical and even from a more translational side, what are your thoughts on that in terms of how do we experiment that?
Yeah, thank you so much. Okay, so we need a ton of money and a ton of animals, which is the answer to like every experiment. But I think you're hitting on a multiple really important points here. The first one is the age of exposure. So for example, some of the best replicated findings about the consequences of THC exposure in humans relates to adolescent exposure and increasing the age of onset for schizophrenia, right? That is one of the most consistent findings. That doesn't happen if you give them perinatal exposure. That doesn't happen if the exposure is during adult, right? So, you know, I think we all know that physiology changes with age, and the way that the systems are functioning, their capacity for change are all going to be different. So I think for our aging study, we really want to push from what would be middle age. So a lot of our animals are like, again, correlating a mouse to a human, like it's hard, but you know, they're like 45 to 60-ish. We want to get those 18 months old. It just takes a long time to prepare them, because I do think that the age dependency of the effect is critical, and it's really important for our guidance. And I think the other point that you make is the chronicity of exposure. So we see deficits induced in cognition in animals that have had like really chronic exposure, meaning exposure in early adolescence, exposure in emerging adulthood, and exposure in middle age. Whereas if we just expose our mice in older age, we get a beneficial effect. So is that because of the age? Is that because of the amount? So what we need to do is parse apart by essentially repeating that same chronicity experiment with our mice as well.
Yeah, exactly.
So I think it's a huge grid of variables, and I think one of the important things, especially when I'm thinking about the translation, is finding out from clinicians and clinical researchers what are the elements that you already know are the key ones, right? Because then I'll do those first.
Yeah, I would definitely say that just finding out the chronicity part would be very critical, and then even like, you know, bringing up the fact that you're kind of like already testing like the type of dosage approach, which is the vapor approach, and how that's detrimental. And it's so I don't know if experimentally can you test that they're exposed to THC without the vapor approach, and what do you see in terms of maybe the chronicity and age interaction? So that would be a very from a more translational side. I think that would be interesting to see.
Yeah, so not just dosage, but mode, right? So
mode and dosage.
Increasingly, there are a number of groups that are actually using edibles. So again, trying to get at how folks are using. And so, you know, we watch what they're doing, a lot of their stuff in adolescence, because they're looking at essentially the risk of having appealing cannabis products accessible to youth. But that formulation would be another great kind of translational partner. I think the other thing, in addition to dose, is we know that THC alone versus with CBD, at least from like a molecular signaling aspect, is different. They're different from one another. The CBD modulates the activity of the THC. And so my graduate student is really interested in bringing in a ratio that we would see like in a commercially available cannabis product, and CBD in isolation, because CBD in isolation is also commonly used. It is the one that, societally we push people to try. You can't sleep, you're anxious, you're not hungry, right? So dose, chronicity, formulation, I think are really important when you're thinking about translation.
Absolutely. Well, thank you so much. That was really interesting.
Yeah, thank you.
Okay, well, if there are no further questions, I think we will conclude today's session. Thank you again, Dr. Penrod, for your time and for sharing your expertise with us today. And thank you to everyone who joined us for today's seminar. Hope to see you again in our future research talk. And well, have a wonderful day.
Thank you. Bye, everyone.
Thank you. Bye.